Chapter 16 is on population evolution. 16, Section 1 is on population genetics, which tells us that we're studying evolution from a genetic point of view. So as opposed to looking more at outside physical features, we're actually studying the genes now. A population, we've mentioned before, but just a reminder, the population is a group of individuals of the same species that are able to interbreed. And a population is going to have some different variations in it. If we were to plot those, they tend to fall in a bell-shaped curve where the average traits all fall in the middle and the extreme traits are these end parts. So the majority of the population has the average trait, and then there's a few of the extremes. So for instance, height, there's a few really, really tall people, there's a few really, really short people, but the majority of the population falls in the average range. And you can pretty much do that with any sort of trait for the population. And the population is going to be the smallest unit that we see evolution occurring. The only thing smaller than that is an individual, and you're not going to see an individual evolve because it doesn't happen within one lifetime. It's something that's genetically based, so it would have to occur over generations as opposed to just within one individual's life. Now, how do we get these variations in a population? There's a couple different ways, which we've talked before about in our genetics unit. They can be either genetic factors or environmental factors. If it's genetically based, then something like a mutation. So a mutation is a change to the DNA, so that's obviously going to give a variety to the alleles that are already present, whether those would be a good mutation or a bad mutation. There's also recombination. We've mentioned crossing over, where the homologous chromosomes exchange pieces of DNA, an independent assortment, where they line up randomly before they're split off into the new cells. So that process also allows for variety. And then the last one is random fusion of gametes, or a fancy way of saying fertilization. So there's hundreds of millions of sperm cells, and only one of those are going to fertilize the egg. So that particular sperm that's carrying those genes is the one that's going to contribute to the offspring. And so the other factors to look at are environmental factors. So things like the amount or quality of food available or space available for habitats. So all of that can provide variety for the population as well. Some definitions you need to know. One is the gene pool, and that's going to be the total genetic information in a population. So every organism that can actually reproduce is going to be a part of that gene pool. Anyone that could potentially pass on their traits. Allele frequency we'll look at, and that's going to tell us how often a certain allele occurs in that gene pool. So you take the number of that particular allele and divide it by the total number of alleles in the population. So if you wanted to track the recessive trait, you would take how many recessive alleles there were over the total. If you want to do the dominant, you'd do it the same way, but number of dominant alleles over the total. A couple other definitions. There's some symbols we'll be using in an equation here in a minute. P stands for the allele frequency of the dominant trait, and Q is the frequency of the recessive allele. And then the last one is phenotype frequency. So instead of just looking at alleles, we're actually looking at phenotypes or appearances. So it's the number of individuals with a particular phenotype divided by the total of individuals in your population. This equation here is called the Hardy-Weinberg equation, which these two gentlemen came up with, and it's p squared plus 2pq plus q squared equals 1, where p squared represents the homozygous dominant individuals, or those that have two dominant alleles. PQ represents the heterozygous individuals in a population, and Q squared represents the homozygous recessive individuals, so those that have two recessive alleles. So this equation can be used to calculate the different allele frequencies. Hardy-Weinberg equilibrium is something that is used when studying populations in order to make things a little bit simple. It's an ideal hypothetical population that is not evolving or not changing. And that just makes things easier to study than something that's constantly changing. So, if a population is at Hardy-Weinberg equilibrium, that means that their allele frequencies are remaining constant as time goes on and that no evolution is actually occurring. This situation will only happen when all 5 of these assumptions take place. So there's random mating, which means that there's no actual selection of a mate based on particular traits. The second one is that there's no selective advantage of a genotype, so essentially there's no natural selection. No one genotype is better for a situation than another. The third one is that no mutations could occur. The fourth is that no migration of individuals could happen, so no one moving in or out of the population. And the last one, you would need to have an infinitely large population size in order to actually have no change occurring at this Hardy-Weinberg equilibrium. So what does this have to do with evolution? Basically, evolution is going to happen whenever this equilibrium is disrupted. So any one of those 5 things gets changed, then that's a disruption to the equilibrium and evolution will happen.